Who cares? All right, everybody, welcome to Grand Rounds. This is today's CME code. Before the first slide, Will was supposed to include a slide saying welcome to all the resident applicants, and I'll be talking to you in a little bit. And, you know, welcome from Durham and Duke University Medical Center. A little bit of background. Today is the birthday of Daniel Webster, Cary Grant, and A.A. Milne, who was the author of Winnie the Pooh. It's a big day in history. In 1803, Thomas Jefferson asked Congress for the money to fund the Lewis and Clark expedition. The X-ray machine was shown for the first time. This coming Sunday starts the Lunar New Year, and it's going to be the year of the rabbit. I don't know how many of you have dogs, but there's my dog, Daisy. She's part hunter, and Daisy thinks a rabbit is like a dog, and that rabbit is not looking at her at this moment, and Daisy's got the drop on that rabbit. But of course, rabbits have 360-degree vision. Daisy doesn't know that, and Daisy has tracked about 100 rabbits and never even got close because the rabbit's staring right after her. I've also heard that rabbits see differently out of each eye, that each eye has slightly different visual properties. And the one focused on Daisy right there is the runaway one, after I eat as much as I possibly can. Today, this is for all of our friends from Michigan. It's National Michigan Day and actually National Peking Duck Day. I actually had a Peking duck in Beijing once, and I had one in Cary, and they were both great. Cary's a suburb of Raleigh. In our research roundup, Danny Laskowitz was the senior author of an article looking at NP 10679, which is an NMDA modulator in acute brain injury. Jamila Minga was the senior author of a study looking at racial and ethnic differences in post-stroke recovery from right hemispherical strokes. Our all-star is Sarah Hassani, an anonymous patient, comment on her being truly excellent. I was looking up Sarah last night for a picture, and I think she's actually a Fulbright scholar, if I'm not mistaken. I mean, that's very impressive.
That's correct.
Is that correct?
Yes.
Very impressive. We used to say there are no Fulbright scholars on the hockey team. Another Duke Neurology all-star, and this one is weird. Shahzad Zafir, a patient, says, "You're a great doctor and I'm going to miss you." So one of the two of them is leaving. Okay, I'll have to dig into that a little bit. In the newspaper yesterday, copperheads in Raleigh in January coming out for the sun. Best advice, leave it alone. Don't poke it, prod it. You know, these snakes live underground, but they're incapable of burrowing. So they have to find spaces that are already there. And then North Carolina is very big on snakes. We've all had snakes in our garages and things of that sort. Again, this is today's CME code. And our case presentation is going to be by Dylan Ryan. Or Ryan Dillon, I'm still not sure which. Got an S. All right.
Yeah, you're good.
Take away.
Cool. So it is Dylan. I go by Rhino as well. If you call me Ryan, it's fine. I'm one of the fourth-year residents here doing a case presentation going back upon the same theme. That sounds like your phone, Rich. Of last case presentation 4 or 5 months ago, kind of going back in the phone booth like Bill and Ted, going back in time.
But yeah, I'm going to plan to go.
So we've got an 84-year-old male who we were consulted on for left-sided weakness, numbness, and slurred speech with concern for vascular etiology of his presentation. Considering his past medical history has a number of vascular risk factors, including hypertension, hyperlipidemia, peripheral arterial disease, and a significant tobacco use history. There's his vitals for completeness, mildly bradycardic, afebrile, statting well. On his initial exam, it was noted he had a right lateral gaze palsy with a right lower motor neuron facial palsy, as well as left-sided upper extremity pronator or left-sided pronator drift with left-sided weakness with full strength on the right, decreased sensation to all modalities on the left in his upper and lower extremity, and right-sided dysmetria on finger to nose. Scoring this, he had an NIH stroke scale of 7 on his initial evaluation. He ends up getting an MRI, and we can see in the right inferior pons, he has an area on DWI of a diffusion restriction with an ADC correlate consistent with an acute stroke. And why this is interesting, if you've been around me, I guess you know that I'm interested in these brainstem stroke syndromes and some of this neurology history. This is what we call inferior medial pontine syndrome, otherwise known, if you Americanize the name, as Fauville syndrome. It was first described by a French anatomist and psychiatrist whose father was actually also an anatomist in the early 1800s, Achille Louis Fauville, back in 1858. He was studying eye movements in the pons and described a 43-year-old male who had a horizontal gaze palsy, a complete facial paralysis, and contralateral hemiparesis, and described this syndrome with this triad of symptoms, essentially. This same triad exists in Millard-Gubler syndrome, but we'll talk about why they're slightly different. These people often have contralateral sensory loss as well and can present with ataxia, a Horner syndrome, or even hearing loss, and we'll talk about why. And the reason why, kind of going through the anatomy and kind of stealing a diagram that took some artistic license, looking at the sagittal and the axial cut, you can see we're at the level of the facial colliculus. You involve the PPRF and 6 nerve nucleus there as the facial nerve kind of comes around the 6 nerve nucleus, creating your lower motor neuron facial palsy. You're hitting the corticospinal tract as it descends in the pons, but also kind of medial there, you have the medial lemniscus with all your sensory information. And that's why you get often the sensory loss with this type of lesion, as well as fibers from the brachium pontus that can cause a dysmetria. Whereas if you look on the sagittal with Millard-Gubler, typically you're more ventral with your lesion and slightly more lateral as you get the nerves coming out of the pons, the 6th and 7th, and the corticospinal tract, preserving so you don't have sensory loss or that ataxia. This gentleman ended up discharged on Plavix. He presented on aspirin. He was switched during his hospital admission. He was already on Crestor 10. He still had an LDL greater than 70. That was increased, did not have evidence of diabetes, did have a positive bubble on his echo. He is 84, but the etiology of his stroke was felt to be small vessel diseased. He was eventually discharged with home health and followed up in our clinic and was doing well. Here are my references. Appreciate your time.
Hi, folks. It's me again. So we're very thrilled that this is the 2023 version of the Jonas Massey Leaders in Neurology lecture series. Jonas is still a very active neuromuscular physician at Duke. Let me just get this out of the way. Jonas began her career as a high school math teacher after graduating from Abilene Christian. Her husband, Wayne, also on the faculty here, went to medical school first. And with Wayne's encouragement, Jonas decided to pursue a career in medical research, then wound up going to Georgetown Medical School after she told me she realized Wayne would come home to do some studying with his friends. And she thought, I'm just as smart as these guys, and went to medical school. In the time since, she's had almost 150 publications and 10,000 citations. She's a world expert in myasthenia gravis and cervical dystonia. The list of awards is summarized very briefly there. In 2022, she won the AAE and M, I don't know if I have that right, sorry, Lifetime Achievement Award. And she's been on multiple boards, and most importantly for us, was the founding director of the Duke Neuromuscular Division and is a great friend and advisor. This is now, I believe, our 7th. This is a list of incredibly distinguished people, starting with Story Landis in 2016. I'm sorry, Lauren. I thought it was Sansing and it's Sansing. I would say Lauren is the only person on this list that I didn't know as a friend before the lecture, which is why I apologize for misspelling your name, but we're thrilled to have you here today. You join an incredible group, starting with Jonas and then all these great physician scientists here. And to introduce Lauren, I'm going to have Brian McCrory come to the podium.
Thanks, Rich. Really appreciate it. It's a great honor to be able to introduce Dr. Lauren Sansing for this really special day in the life of the department, the Jonas Massey Lectureship. Dr. Sansing joins us from Yale, where she's the academic chief of the Division of Vascular Neurology, and she serves as vice chair for faculty development at Yale. Dr. Sansing really kind of has a global reputation based on her achievements in translational research, clinical care, and leadership in the field of neurology and vascular neurology in particular. Her lab has been responsible for a number of really seminal discoveries in the field of translational ICH research, including demonstrating for the first time that monocytes infiltrate the brain during ICH, differentiated to macrophages, revealing a role for TGF-beta in suppression of microglial activation in ICH, and then most recently, and hopefully without giving too much away before the talk, a really interesting characterization of the different sort of layers and stages of the immune response in humans during ICH based on a really kind of inventive sub-study of the Misti III trial. She's got a number of awards, Best Doctor in America for the last 10 years and counting. She was recently inducted into the American Society of Clinical Investigation, and she occupies a number of very senior leadership positions, including directing the NIH-funded Stroke Preclinical Assessment Network for Acute Neuroprotection and the Buger Center of Excellence in Hemorrhagic Stroke Prevention and Treatment. She's a very dedicated mentor, particularly to sort of trainees and junior faculty, and I myself can attest to that personally. She's mentored dozens of junior researchers and inspired and enriched the careers of many, many more. And so we're very grateful that Dr. Sansing has taken the time out of her schedule to visit us today. So thank you, Dr. Sansing.
So it is really my honor to be here as one of the Massey lecturers. Indeed, Dr. Bryan, you pointed out what an incredible list of prior lectures you've had. And so it's an honor to be here and among that list. And thank you, Dr. McGrory, for that really kind and way too generous introduction. So I'm going to be talking about inflammation, neuroinflammation after intracerebral hemorrhage. And as you just heard, I'm a stroke neurologist. I'm also scientifically an immunologist. And so my lab spends its time trying to dissect the immune responses in the brain, moving between mouse and human. So we spend a lot of time in mice, but our overall goal, of course, is to find translational targets for humans. And so I'm hoping that there's Yep. MD is not a mouse doctor. So we spend a lot of time doctoring mice, but our goal is to treat patients. I don't have any relevant disclosures. So intracerebral hemorrhage, just to bring us all onto the same page, is when a blood vessel within the brain ruptures, and it leads to a mass of blood within the brain parenchyma itself. And not surprisingly, this triggers a robust inflammatory response. It only accounts for about 15% of strokes in Western countries, more in Asia, but it's responsible for about 40% of all stroke deaths, and only about 20% of survivors ever reach functional independence. I think about ICH as a process. So the initial event, after years and years of disease pathophysiology that leads to weakness and fragility of the blood vessel wall, is that there's the blood vessel rupture. This leads to early hematoma growth. We know that about 25% of the patients are still bleeding when they reach our ER, higher numbers the earlier they present. And this hematoma enlargement leads to tissue disruption and shear forces on the white matter tracts in the brain. There's then a robust triggering of the inflammatory response. This is characterized by cytokine and chemokine, those proteins that signal to other cells to activate them or call them into the brain, microglial activation, and robust infiltration of blood leukocytes into the brain, the perihematomal region. This initially is neutrophils and NK cells, leads to monocytes, then followed by T cells, and maybe even later B cells. Over days, red blood cells lyse within the brain. They release hemoglobin, which is then degraded and releases toxic iron. And this leads to another cascade of oxidative injury, triggering again an inflammatory response. And this process goes on for days. Over time, there's phagocytosis of the hematoma and all those red blood cells and the other debris within the brain, secretion of growth factors, and hopefully resolution and repair. And all of this is under the umbrella of inflammation. So there's a huge need for translational studies to understand what are the targets that can be modified to improve outcomes in our patients. That's my doctor hat. And then this is a complex inflammatory process within the most complex organ. So super exciting to study from a research side. I'm going to focus on macrophages for most of this talk. And so just to make sure that when I say the word macrophage, we're all on the same page, there are two main macrophage populations within the brain in the context of iceage. So the first is the resident microglia. These are seeded into the brain during embryological development. They come from the yolk sac. Under homeostatic conditions, they're a self-renewing population, long-lived, radio-resistant, but very plastic. Can activate, secrete pro-inflammatory cytokines during homeostasis. They prune synapses and sort of survey the brain, secrete growth factors like BDNF, and are really a critical macrophage population in both surveillance and response to injury. The other macrophage population that I'm going to talk about are the monocyte-derived macrophages. So these exit the bone marrow as classical and non-classical monocytes in a CCR2, CCL2 dependent manner. They circulate in the blood, and then in the context of injury or inflammation, they cross the blood-brain barrier and traffic into the brain, differentiating into macrophages. Hemorrhage, and there they go. There are other macrophage populations in the brain. There are perivascular macrophages, there are meningeal macrophages, choroid plexus macrophages. I'm not going to talk about them because they're in smaller numbers and we don't really have good phenotypic characterization of them yet. So when I say microglia, I'm talking about those resident macrophages. And if I say macrophages, I'm talking about monocyte-derived macrophages. So my lab started out at a time where macrophage responses were really characterized as M1 and M2. So M1 being pro-inflammatory, secreting IL-1 beta, TNF, IL-6, and M2 being involved in brain tumors, alternative activated macrophages in response to allergy, helminth infection, and really driven by sort of IL-4 and IL-10 responses. We couldn't find any IL-4 or IL-10 in our models, and we were trying to figure out what are the sort of phenotypic variabilities of the macrophages within the brain. And are they really good and then bad, or are they mixed all the time? And so we took a very reductionist approach and decided we're just going to deplete them at different time points after mouse hemorrhage and see, are the mice better or worse? And that'll give us a broad idea of what are the stages in which they're contributing to injury versus recovery. And because my lab works in both mice and humans, if you're lost, look at the upper right-hand corner of the slide. If it's mouse data, there's a mouse, and if it's human data, there's a little man. So in the first stage of the experiment, we took an acute depletion model. So we used an anti-CCR2 antibody, so this will deplete all circulating macrophages, but not the microglia, versus an isotype control. We induced ICH by injection of collagenase to induce bleeding within the brain. And then we did behavioral testing for the first couple of days. And what we found is that when we subjected the mice to a large number of neurological tests, on every test, the mice that did not have macrophages that were treated with this anti-CCR2 antibody, the gray triangles, were protected from a neurological deficit. So they were better on using their left forelimb than the mice who had normal macrophages, suggesting that the macrophages during these first 3 days are contributing to that neurological disability, because if you don't have them, the mice look much better. However, if we induced hemorrhage and then allowed normal macrophage recruitment to the brain for the first 3 days, and then randomized the mice to either the CCR2 depletion antibody or the control, and then did behavioral testing out to the second week, we found a very different effect. So here, if you can see, at day 12, there's more residual hemorrhage left in the brains of the mice that didn't have macrophages, and this is quantified here. And on the various neurological tests, the mice that had normal macrophage recruitment in the black circles had normal neurological improvement over time, but the mice that did not have macrophages failed to improve, shown on both the cylinder test and the corner turn test. So this shows us that from that somewhere in the 3 to 5 day time point out, the macrophages are contributing to recovery. They're phagocytosing the hemorrhage or doing something that are inducing other cells to phagocytose the hemorrhage, and they're contributing to neurological recovery. And I mentioned that some graduate students in my lab had spent a lot of time trying to find IL-4 and other alternative mediators and anti-inflammatory cytokines unsuccessfully. So we took a step back and we cell sorted the macrophages from different time points after hemorrhage and then transcriptionally profiled them. We also looked at phagocytosis specifically in the macrophages and the microglia to try to understand which of these cell types are really doing the bulk of the clearance after ICH. So mice were induced with hemorrhage. This is now by autologous blood injection. We labeled the red blood cells before we injected them so that we could track them by flow cytometry after we sacrificed the mice. And we also did some experiments where we took the macrophages and the microglia and put them into cell culture with phagocytosis beads so we could quantify how robustly those cells took up the beads. So this is an image using the Abnis image stream. It's a flow cytometer, so it can track the sort of expression of different antibodies on each of the cell and quantify them as they run through the cytometer, but it also takes a picture of every single cell as it goes through. So you can see the microglia here in the top row. Here's the bright field image, the fluorescent bead. It has 1 bead in it. It's low on CD45, the leukocyte marker. It has a nucleus, a DAPI, and it does not express Ly6C or and low CD11B that doesn't project particularly well. The monocyte-derived macrophages, on the other hand, are engulfing more beads here. They're higher on CD45. They also have a nucleus. They're high on Ly6C and CD11B. So this shows us sort of visually that indeed we can find our cells, they're nice, they're healthy, they're live, our markers are showing what they should. And when we quantified it, the macrophages were engulfing a lot more beads than the microglia. But that's beads. So we decided to go ahead and look at day 3, the microglia and the macrophages engulfment of red blood cells, a much more physiologically relevant substrate. But we found the same thing. So when we quantified a number of mice at day 3, looking at their macrophages and their microglia, we found that indeed the monocyte-derived macrophages were much more robust in clearing the hemorrhage, clearing specifically the red blood cells from within the brain. So then we took similar mice. Now this was induced by collagenase injection and did that transcriptional profiling that I mentioned just a moment ago. So we sorted the macrophages from within the brain. We had to pool 3 mice per experiment and then replicated this 3 times per time point and looked at them at day 3, day 1, day 3, day 7, and day 10. After day 10, we couldn't find a lot of macrophages within the brain. We transcriptionally profiled the populations by a custom nanostring panel at this point, which was 780 immune-related genes. And here's the data just summarized in a heat map. You can see robust gene expression. So redder on the heat map is higher gene expression in genes down here at day 1. At day 3, a different set of genes turns on. And then we were most interested in these genes here that were turning on at day 3 as the mice were beginning to recover, but then robustly expressed during that entire recovery phase. So what are these genes? The bottom are classic innate immunity pathways, NF-kappa B, IL-1 beta, TNF, MIF, HIF-1 alpha, and class 2. And here, not anti-inflammatory cytokines, but genes involved in efferocytosis and the resolution of inflammation, like AXL and annexin A. Phagolysosome formation, so consistent with this data that we had generated that the macrophages were phagocytosing a lot of debris, as well as cholesterol transport and efferocytosis, as well as lipid efflux. So this efferocytosis pathway really piqued our interest. And the number one gene that was upregulated at day 7 compared to day 1 was this gene called AXL. So AXL is part of a family of efferocytosis receptors. These receptors bind phosphatidylserine on the external leaflet of cell membranes that are undergoing apoptosis. So when phosphatidylserine gets translocated and now is expressed on dying cells by apoptosis, AXL, as well as TYRO3, MERTK, and a number of other phagocytosis or efferocytosis receptors will bind to it, allowing the engulfment of those dead cells, which induces a reduction of inflammation in the macrophages, as well as the ability to phagocytose more dead cells. This has been studied in wound healing and a number of other processes prior to our work, but had not yet looked at this process in intracerebral hemorrhage. This was a nice tie between wound healing responses known in the systemic immune system and resolution of inflammation after iceage. So we wondered whether indeed this was the process going on in our mouse model. And we showed in cell culture that indeed mouse macrophages gobble up red blood cells that are expressing phosphatidylserine, and this reduces inflammatory cytokine production by those macrophages. So then we switched to human cells and created monocyte-derived macrophages in the human cells, and showed indeed that if we could induce phosphatidylserine by heat shocking them, that's the HS, the gut macrophages in green loved to gobble up those red blood cells. So normal red blood cells in red, macrophages in green here, and you see some engulfment. When you induce phosphatidylserine expression on the red blood cells, there's a much more robust uptake. And if you block that phosphatidylserine engagement using annexin 5, you can reduce the phagocytosis. Indeed, we showed that this engulfment of the phosphatidylserine expressing macrophages, red blood cells, induced hemoxygenase expression, which is an antioxidant, and reduced TNF production by the macrophages themselves. So phagocytosis of the red blood cells by efferocytosis directly modulated the macrophage phenotype, linking the reduction in inflammation with the clearance of the hematoma in a single pathway. So we were very excited. We went back to the mice, used AXL and MERTK double knockouts, showed indeed there was more residual blood left in the brain in the AXL and MERTK double knockouts, and they had worse neurological outcomes out in the subacute phase, shown here at day 3 and day 7. So I showed you a bit of the sort of mouse and human approach that we took in identifying one pathway that can reduce inflammation and clear the hemorrhage. We had worked in some other mechanisms earlier, had shown that macrophages are coming into the brain in a CCR2, CCL2 dependent manner. They're using alpha-4 integrin because if we blocked that integrin, we could block macrophage recruitment. That they were very pro-inflammatory early in the first couple of days and contributing to neurological disability. And then through this process of efferocytosis, they're reducing the sort of phagocytosing the hemorrhage and also reducing their pro-inflammatory phenotype. So we have this nice model now where the macrophages in the brain are transitioning phenotypes over time. And we had mapped out that time period in the mouse model where they were pro-inflammatory for about 3 days and then anti-inflammatory for the second week. And we showed that efferocytosis is the mediator for the macrophage transition. And in data that I don't have time to show you today, we show that the microglia are also very pro-inflammatory early, making a lot of TNF and CXCL10. And then in response to TGF-beta, transition their phenotype to make more BDNF, reduce their pro-inflammatory cytokines, and particularly IL-6 production, and contribute to recovery. So with this background, we turned our attention to the Misti III clinical trial. Misti III was evaluating in a very large phase 3 international trial the efficacy of minimally invasive surgery to improve outcomes after hemorrhage. So patients with large, greater than 30 cc hemorrhages were consented and enrolled into this clinical trial. A catheter, if they were randomized to surgery, a catheter was placed within the long axis of the hemorrhage. Here's our first patient at Yale, and you can see the catheter here. And then over the next several days, TPA was injected into the catheter to liquefy the hematoma, and then it was allowed to drain out by gravity. So this is a gentler, minimally invasive surgical approach for evacuation of the hemorrhages. And as I mentioned, Misti III was trying to figure out whether this was better than best medical management. But all the blood and hematoma was being thrown away, as is often done. And so we launched a sub-study called iceage. At 16 sites around the country, patients had their daily hematoma evacuate and a matched peripheral blood sample collected, immediately placed at 4 degrees, FedExed overnight to our lab, where we then cell sorted the populations of interest and transcriptionally profiled them by RNA-seq. This work was led by Mike Askinase and Britt Goods, who did an amazing job with a whole lot of troubleshooting to get this study to be a success. We found we were able to generate 140 monocyte profiles and 125 neutrophil profiles, ranging from about 37 to 171 hours after symptom onset across 22 patients. So I'm going to show you data now grouped by time point, because what we found was that there were 2 distinct phases of macrophage as well as neutrophil activation within the brains. There's an acute stage that lasts for the first 4 days, where there's robust and dynamic changes in transcriptional activation. And then from about 4 to 7 days, there was a more stable phase of activation where they were activated, but they just weren't changing very much from time point to time point. So here in these heat maps, I'm showing you just some cytokines, chemokines, and some eicosanoids in the macrophages and neutrophils from the brains of the patients relative to their expression levels in blood. So if the dot is red, it's much more robustly expressed in the brain compared to the same cell population in the peripheral blood. So just look at the big picture. There's robust activation of cytokines in the macrophages as well as chemokines. And then there's robust activation of prostaglandin E synthase, which is the enzyme that makes prostaglandin E2. I point that out because it becomes important later on. Interestingly, what we found also was that there was robust activation of genes involved in glycolysis. So as you all remember, glucose is taken up into the cell through glucose transporters. It then undergoes sort of enzymatic use in glycolysis and then enters the Krebs cycle to generate high levels of ATP during homeostasis. But what had been known and discovered in some tumor and infection models was that cells, and particularly macrophages, during acute inflammatory stimuli have breaks in the TCA cycle, and they're really much more robustly utilizing glycolysis, allowing for some of those intermediates in metabolism to be shunted to other cellular functions, like making cytokines, making reactive oxygen species, and building blocks for cell proliferation. So we found that the glycolytic enzymes expression was much higher in the hematoma macrophages compared to the monocytes in the blood. And doing pathway analysis, the #1 pathway that was upregulated in the hematoma wasn't inflammation, but it was glycolysis. And here's just a summary of what I just told you. When you stimulate macrophages with pro-inflammatory signals, they upregulate glycolysis and downregulate cellular respiration. If you stimulate those macrophages with IL-4, an anti-inflammatory signal, they increase cellular respiration and fatty acid oxidation, and they make an anti-inflammatory macrophage. So then this didn't really surprise us that the macrophages were, during that acute phase, were really utilizing glycolysis. And what we found is that when we looked over time, macrophages indeed, in gold from the hematoma and in blue from the blood, decreased production of IL-6 and IL-1 alpha. They maintained production of anti-inflammatory mediators like IL-10 and prostaglandin E synthase, and they upregulated phagocytosis markers like MERTK, like I had mentioned earlier from our mouse work, and hemoxygenase 1, which helps them handle all that red blood cell oxidative stress. And when we looked at what were the dynamic changes in the macrophages, we found that HIF signaling, so HIF, hypoxia-inducible factor, is a major driver of glycolysis, as well as TNF and other inflammatory signals were decreasing over time. And increasing were things like phagosome and lysosome processes consistent with the mouse data that the macrophages in the brain were becoming less pro-inflammatory and they were phagocytosing debris over time. So these were, wonderful sanity checks that in the human brain, the living human brain, the same processes that we were studying in the mice were actually happening. And interestingly, the time course roughly matched up. And because this was a clinical trial, we had robust outcome collection and we had robust clinical data. And so we were able to look at differential gene expression in the hematoma of the macrophages and good clinical recovery out as far as 1 year in the Misti trial. We used the same modified Rankin scale score that Misti used. We used the same controllers adjusting for iceage severity as Misti did. In our neutrophils in the blood and in the hematoma, we found no differentially expressed genes. That doesn't mean that neutrophils aren't incredibly important. It just means transcriptionally, we couldn't find those important factors. But in hematoma macrophages, we found 561 differentially expressed genes that contributed or were associated with good outcome. So I brought up the whole metabolism side story a few minutes ago because it ends up being critically important here. We thought because pro-inflammatory macrophages are associated with utilize glycolysis, in fact, they have to utilize glycolysis, that the patients that decreased glycolysis in that subacute phase would have better recovery, right? They would be turning off their pro-inflammatory state and they'd be upregulating OXPHOS and fatty acid metabolism. It was actually the complete opposite. So the patients that had good outcome here upregulated the critical genes in the glycolysis pathway, downregulated the genes involved in the TCA cycle, and upregulated immune factors like prostaglandin E synthase. And in fact, when you do pathway analysis, looking for those pathways that were associated with good or excellent clinical recovery in the patients, it was HIF and glycolysis that correlated with good clinical recovery. So what is prostaglandin synthase and prostaglandin E2 and what's known about it in inflammation? In a model from Yasmin Belkade's lab at NIH, it was shown that neutrophil TNF production was reduced by prostaglandin E synthase and improved gut inflammation. In a model of ICH in Ratan's lab, it was shown that PGE2 reduces neuronal ferroptosis in the presence of hemorrhage. An EP4 agonist, so directly activating the receptor, reduced blood-brain barrier permeability and inflammatory cytokines in ischemic stroke. And in a model of neuronal inflammation, EAE, a model of MS, stem cell production of prostaglandin E2 directly reduced inflammation and improved outcomes. So this link between prostaglandin E2 and good outcome was really tantalizing to us. And yet we didn't understand the connection between the metabolic reprogramming in the macrophages and the production of this anti-inflammatory mediator. So we turned to cell culture again and used human monocyte-derived macrophages. We created a stimulation cocktail that we call iceage damp. It has some immune factors and some thrombin, IL-1 beta, and a factor called S1A8 that is produced from neutrophils upon degranulation. It's the sort of one of the top proteins in neutrophils. And we wanted to sort of not use LPS, right? When we do immunology research, we often use LPS and interferon gamma to make pro-inflammatory macrophages, but those factors aren't in the brain after ICH. And so we needed to come up with a cocktail that more closely mimicked what was happening in our patients. So we developed this cocktail, showed that indeed, if we stimulate the macrophages with this damp, we get robust production of IL-6, but we also get robust production of these anti-inflammatory mediators like PGE2 and IL-10. we showed that this was directly dependent on glycolysis, because if you blocked glycolysis with 2-deoxyglucose, that's 2DG here, you got a reduction in IL-6 production, as well as a reduction in PGE2 and IL-10. And if you directly inhibited HIF with akinomycin, you got the same result. You got a reduction in IL-6, as well as a reduction in PGE2 and IL-10. So this showed that this glycolytic metabolism driven by HIF was necessary for the production of both pro-inflammatory mediators like IL-6, as well as anti-inflammatory mediators like PGE2 and IL-10. And then we took the healthy donor macrophages, stimulated them with this damp cocktail, with or without the direct application of PGE2. And what we found is that with PGE2 addition, each individual person's donor macrophages reduced IL-6 and increased growth factor like VEGF secretion in the macrophages, finding that PGE2 can directly modulate the phenotype of those macrophages in culture. So what have I shown you here from the human cells? What I showed you is that the myeloid cells, the macrophages and the neutrophils, engage in this conserved two-stage transcriptional response. And in the interest of time, I didn't show you how consistent those responses were across patients and a number of other sort of quality metrics. There's this acute phase that lasts for about the first 4 days. Now, the caveat there is that we didn't have the first sort of 24-hour data, so I can't tell you what's happening during those first 24 hours just because of the need to have hemorrhage stability in the Misti trial. But at least from those sort of 2 to 4 days, the macrophages are dynamic within the brain. They're changing rapidly hour by hour. And then there's this subacute phase from about day 4 to day 7, where they're activated in the brain, but they're transcriptionally not as dynamic. During that time period, there's a decrease in pro-inflammatory gene production, there's an increase in phagocytosis gene production, and an increase in hemoxygenase 1 and prostaglandin E2. Luckily for us, and thankfully, in terms of translational research, the time periods for these changes in humans lines up nicely with what's happening in mice. We confirmed the prevailing view that glycolytic metabolism is critical for supporting and is necessary for pro-inflammatory features in macrophages, but we showed it in vivo in the brain in humans, which was very fun. And then surprisingly, and this is where our hypothesis was completely wrong, sustained glycolytic metabolism is associated with and necessary for PGE2 synthesis and good neurological outcome in the patients long-term. And that the anabolic metabolism driven by HIF was required. So that was bulk macrophage sequencing over time using the Misti clinical trial infrastructure. We also wanted to understand macrophage heterogeneity at the single cell level. So we took the first patient that we had extra sample from that iceage-seq sub-study at all the time points in which we collected data from that patient and analyzed the cells using SQL for individual cell heterogeneity. Here's just the overall UMAP. On the bottom left is each individual cell colored by whether it came from the hematoma or the peripheral blood in red. Here it is colored by hours after onset. And so you can see that the cells are indeed grouping or clustering by the time period from hemorrhage onset that the sample was collected from. And then here it is colored by cell identity. And we have lots of T cells, macrophages in purple, which I'm obviously going to zoom in on, NK cells, CD8 T cells, B cells, and a small group of neurons. So I'm just going to show you the macrophage population, and in particular, the macrophage population in the brain. Here it is subclustered, where you see the cells that came from the hematoma in gray and the blood in red. What I will say is that this patient had a slight rebleeding event. It was asymptomatic. The catheter was open, but the patient started oozing and draining more blood during that time period. And then it stopped as we stopped TPA. When at the time period of this bleeding event, you actually get the identification of some more blood-like, peripheral blood-like monocytes and other cells into the hematoma. But even without that, we were able to find this direct sort of and temporal shift in the dominant phenotype of the macrophages within the brain. So that's shown here. In that initial time period at hour 51, almost all of the macrophages within the brain clustered together in myeloid cluster 4. In cluster 4, the number one pathway, not surprisingly, from the bulk data I showed you a little while ago, was glycolysis. Starting at about 73 hours, the patient's macrophages were dominated by cells in myeloid cluster 2. These were characterized by granulocyte and other cell leukocyte adhesion and mobility, diapedesis within the brain. And then beginning at about 89 hours, you start to see the emergence of this myeloid cluster 5 that from over the next several time points really dominates the macrophage populations. And this cluster 5 is characterized by senescence, so the sort of aging of the cell and decrease in its activation, as well as phagosome maturation and growth factor secretion. So here at the single cell level, what we were able to find is that even as the overall macrophage population is going through these dynamic shifts, what that's comprised of is the emergence of different clusters of cells that become dominant at different time periods that have very different phenotypic activation states. And they go from glycolytic and highly pro-inflammatory to engaging in phagocytosis, senescence, and growth factor secretion. So we've continued on this line of evidence, and I mentioned that that was the kinetics in one single patient. We now have about 10 patients that we've analyzed at different time points. And these have been analyzed by the 10x platform, so we have a lot more transcriptional depth. We find robust myeloid populations. We also see neurons and oligodendrocytes, B cells, T cells, and NK cells within the brain. On the right panel, they're colored by blood in red or pink and hematoma in blue. And there's some nice sanity checks here. The neurons and oligodendrocytes, as well as microglia, are only coming from the hematoma, thank goodness. But the other leukocyte populations are coming from both hematoma and peripheral blood, so we can analyze the differences. We've done some analysis of this data. We've done some module scoring looking for HIF targets across the different cell populations. And what we found was that indeed the macrophages are much more enriched for this HIF glycolytic program than the T cells are or other populations. It's not universal. There are some subclusters of macrophages that are very glycolytic and others that are not, and that's shown here. And now we've taken this data back to the murine model and used some conditional knockouts to knock out HIF in the macrophage populations and then create hemorrhages in those models. We've also embarked on some studies of sex and age differences in the mouse model. And this is critically important because there's a lot of translational barriers in hemorrhage and stroke research. And one of them is thought that we're not really capturing the biological diversity in our patients that we do in our mouse models that we have in our patients. And so we've been looking and finding some really interesting upregulation in numbers of microglia in the young and upregulation of macrophages shown here in the aged. And we've found that the aged mice in both males and females are upregulating certain gene transcriptional programs. And there are others that are really only seen in aged females. In particular, the type 1 interferon response is very robust in aging, and in particular in aging females in the microglia. And we see almost no upregulation of this response in young male and young female mice after hemorrhage. Interestingly, this interferon response is also seen in the sham mice, but only in aging females, suggesting that aging female microglia are starting to upregulate interferon responses that may prime them for more robust neuroinflammation. In the macrophages, we saw some similar things, although the responses are a little bit more distinct by sex, where we see an aged male response, an aged female response, and a young female response. And again, we see a strong type 1 interferon response in the aged females. So I'm going to stop data sharing there. And this is a very wordy slide, but it's really hoped to provoke some questions for you. And there are questions that we think about in lab. What does this mean for patients? We've shown now over about 15 years of work that monocyte-derived macrophages contribute to recovery, definitely in mice, and it's highly associated in that transcriptional data in humans. So what does this mean? Blocking leukocyte recruitment, in my mind, is unlikely to help because we really need to harness that reparative macrophage response and not just block all macrophage responses. There are 4 days of rapidly changing inflammatory responses within the brain. So this is exciting, right? 4 days of changing responses means that we likely have a therapeutic window and that perhaps an intervention that speeds up this acquisition of this more resolvatory or reparative macrophage phenotype may enhance recovery by diminishing that early pro-inflammatory stage and enhancing phagocytosis. That's untested. I think that modulation of macrophage metabolism to augment these anti-inflammatory factors, enhance phagocytosis and inhibit iron-induced death, ferroptosis, may be a key therapeutic strategy. And we wonder whether this age-related type 1 interferon response that we're picking up in the mice has implications on recovery or cognition, or the higher prevalence of mood disorders and cognitive dysfunction in older females. And then what about our interest in surgical evacuation? I think if we can find an effective method for getting most of the blood out of the brain, that it'll likely translate to improved clinical outcomes. The Misti trial's post hoc analysis showed that indeed, if you reach the surgical endpoint, patients recovered better. But I think that even if you remove the blood, there's still going to be a robust inflammatory response. It will be better if you can remove a lot of those stimulations, right? So if you get a lot of the necrosis and the apoptotic cells and the debris in the blood from the brain, you'll have a less robust inflammatory response. That's untested. But I think there's still going to be a lot of debris there and a lot of other cleanup for the immune system to do. And there's going to be the need for these reparative responses. It also opens up the opportunity to directly instill a therapeutic agent into the brain because you'll have access, right? You'll have a minimally invasive surgical approach that removes the brain hemorrhage, and then you can instill a therapeutic. That's my dream. So what I've shown you is that we've moved from making a hypothesis in mice and testing it to observing and interrogating the immune system in the human brain. We then take these back and utilize the genetic tractability of mouse models as well as in vitro assays to sort of find the mechanism, test out candidate therapeutics, and understand the pathways better. We then want to look for evidence that those pathways are engaged in our patient samples and that the mechanisms are conserved. We want to test those therapeutics back in mice, but define our therapeutic windows based on the human data. Also, we make the lab very dizzy, moving back and forth between mouse and human, but I think everyone is engaged because they see the therapeutic potential for that work. A lot of the PhDs and other students will come and rotate on, after doing HIPAA training, of course, on service to see the patients that we're trying to help. And this brings a new enthusiasm, right, to sometimes grueling computational and experimental mechanistic work. So with that, I'm going to thank the lab and all of our collaborators. I highlighted some of Mike's work and Britt's work in the iceage-seq data. Feng Chang led the efferocytosis work. John, Connor, Yan have contributed to a number of studies, as have our neurosurgical collaborators. All the Misti patients, investigators, and their families that consented to giving us those samples, and of course, our funding. And with that, I'm happy to take questions.
Thank you very much. We have plenty of time for questions. Fire away.
That was a great presentation. I have a question regarding the macrophage microglia subsets. Microglia are thought to be just the primary surveillance mechanism in the brain, helping to just pick up debris. I just wondered, have you tried like a chimera experiment in which the bone marrow is ablated and you replenish the bone marrow with CCR2 deficient monocytes and see if you actually have the same effect? That's one question. Second
Yes.
is, have you looked at different patients who may have different levels of CCR2 to see if they have differences in terms of hematoma resolution?
Yeah. So those are two great questions. First is whether we've used bone marrow chimera experiments to try to isolate the CCR2 deficiency to the peripheral immune system and knock out the ability for monocytes to traffic to the brain. And indeed, we did. And I didn't have time to show you that data, but it's very consistent and it's in fact even more robust than the CCR2 and depletion antibody data that I did show you, where they have delayed hematoma resolution in the subacute phase, but they're robustly protected from early injury and early disability in the first couple of days. We've also done the sort of swapping experiments looking at CX3CR1 deficiency in microglia and mapped out the kinetics of microglial activation. The microglial activation kinetics are a little bit different, but those bone marrow chimera experiments that you asked about are fantastic tools to really separate out the microglia from the macrophages. In terms of soluble CCL2 levels and outcomes after hemorrhage, it's a little bit more complicated. What we did was we looked at individual levels of a whole bunch of cytokines and chemokines at both the acute phase and the subacute phase. And we didn't find as much of an individual time period association with outcome. But if you looked at what those chemokines were doing over time, meaning were they increasing over time or decreasing over time, that's where we found really robust associations with outcome. And this was in the peripheral blood. And what we found was that patients that were increasing CCL2 levels from 24 to 72 hours had much better outcomes than patients that were not. And this goes along with this idea that if you can continue to recruit macrophages during that subacute phase, when they're being helpful, you would likely contribute to recovery. I didn't have hematoma quantifications in those patients, long-term quantification of how big their hemorrhage was. I just had modified Rankin scale score neurological outcomes. So I can't address sort of the intermediate biomarker of hemorrhage size in those patients that had higher CCL2 levels. But those are great questions. Thanks.
All right, we'll take one from the internet. Wayne, do you want to ask your question? We've had a little trouble with the audio from outside, but take a shot at it.
Sure. Thanks a lot. Great talk. Clear, strategy to reduce inflammatory response at a good target. Corticosteroid has been used for iceage with variable response. The question for you, do you think that big enough? Those strategy would be, big enough to change 1 point on a modified Rankin scales.
Yeah, I think that's a really great question. So the question for everyone, in case you couldn't hear it here in the room, was that do you think that strategies to reduce neuroinflammation is going to be robust enough to actually change a point on the modified Rankin scale score? So we use this modified Rankin scale score that goes from 0 to 6 to measure neurological disability in almost all stroke trials. But you can imagine that that's a really coarse scale, and yet that's how we define efficacy of a treatment. So we have to move people by a point to be able to show a difference. I am biased here because I think that the primary mechanisms after the mass effect from the hemorrhage that induce injury are inflammatory. Of course, I'm an immunologist, but I'm quite convinced of that. When I look at the transcriptional profiling, what we see are immune pathways and metabolism pathways. And so, I think we have a really great opportunity if we can find the right target. And I don't think it's corticosteroids. I think that those are too blunt a tool. But I think if we find the right target that can really manipulate the immune cells to be more pro-recovery and to reduce ROS production and oxidative injury, that we have a real strong chance. Will that translate is the $1 million question. We have to test the target first and obviously in the animal models, but I think we're not that far off. And there are some great trials that are going on looking at different anti-inflammatory mediators across the spectrum, looking at ApoE has come out of lab work here at Duke, as well as looking at a small molecule that'll decrease pro-inflammatory cytokine production that's being tested in the BEACH trial. There are 2 trials that are looking at IL-1 receptor antagonism to try to block that pathway. So I think there's a lot of enthusiasm and a lot of hope that ND will be able to move those patients, but we don't have that data yet.
Lauren, I'll take the last question here, just because you mentioned it and you're at Duke. I've read some of the literature on ApoE and intracranial hemorrhage outcome, and it seems to be kind of all over the place. What's your take on that?
So I mentioned in the macrophages that one of the major genes that are being upregulated in that subacute phase when the macrophages are being helpful is ApoE. So I think the lipid handling pathways are also critically important in reducing inflammation in our models. And I didn't show you some of that data, but there's robust effects of lipid handling and interference with lipid handling and transport on the immune responses, both in vivo and in cell culture. So I'm intrigued by the ApoE story and the ApoE mimetic, and I look forward to the results.
Think of the data that's out there. Does it really suggest that it has an effect in humans on recovery or severity?
Yeah, so the data that was published looked at the very similar cohort from the ERIC trial, and it was very promising. Granted, it's a small study, and against a concomitant different cohort study, but that was well matched and it was promising. So I look forward to these randomized double-blind direct comparison trials, and hopefully they move forward rapidly.
All right. Well, again, thank you for our guests. A big hello to Jonas, who was on the talk. I'm not sure if she still is. Jonas, we're having a hard time hearing in here from outside, so I just want to send thanks and a hello, and to everyone else, have a great day. Thank you.
Thank you.
Yeah, no.
Oh, there she is. You don't know how much pressure that is. This.
Ryan, I hear you laughing from the
Yeah.